Hi everybody, my name is Aviv and I'm a PhD candidate supervised by Professor Micha Spira. The title of my project is Analysis of the Interfaces Formed between Cortical Implants and the Parenchyma, Feasibility Assessment towards Multi-Site Intracellular Recordings. I would like to say first that my work has been published in 5 papers. I will only present the highlights of it. If you will find some part interesting, it is all published in details, and these are the names of the papers. So if you find something particularly interesting, you are invited to read them or ask me. To clarify the motivation of the study, I will start with some background. Among many tool types developed to investigate the brain, electrophysiology holds the most suitable spatiotemporal resolution. In the single to tens of neurons scale, the most commonly used technology to record electrophysiological signals from the live brain is the multi-electrode arrays, for short MEA. You can see here some examples for the standard MEAs that were developed over the years. These MEA were developed to enable long-term multi-site parallel recordings from hundreds to thousands of neurons. But the current MEA technology suffer from two major problems. They can only record the extracellular field potentials and are blind to synaptic potentials and subthreshold membrane oscillations. Therefore, the use of conventional MEA cannot provide all the information we need to understand the brain computation. And the second problem is that the commonly used technologies that exist to record or stimulate the live brain suffer from low signal-to-noise ratio, poor source resolution, and deterioration of recording quality and yield over time. The other type of recording is intracellular. This enables us to record the entire electrophysiological repertoire of a single neuron. This includes the synaptic potentials and the subthreshold membrane oscillations. These signals are usually recorded by a sharp electrode or a patch clamp that demands delicate manipulations and penetrating neuron cell membrane, therefore suffers from substantial time and single neuron at a time limitations, and that is very difficult to apply in a freely behaving animal. Combining the advantages of both intracellular and extracellular recordings, and in other words, a method that enables recording of the entire electrophysiological repertoire of many single neurons in parallel for weeks, months, and years in a behaving animal into one method will be the dream neuroprobe and will substantially enhance brain research and understanding. Despite the limitations that I described now, most of the attempts by the neuroengineering and users community focus on how to increase the number and density of electrodes that a platform can carry, and not how to improve the recording quality and yield. Confronting this issue, a few years ago, our lab have developed an approach enables to record attenuated intracellular recordings termed in-cell recordings by extracellular three-dimensional mushroom-shaped microelectrodes. This is achieved by a configuration in which a neuron, here in pink, engulfs the three-dimensional electrode, here in yellow, forming a high seal resistance between the neuron's plasma membrane and the electrode, and increased conductance of the membrane that faces the gold mushroom microelectrodes. This configuration is very similar to the configuration of a perforated patch electrode, and this configuration allowed us to record in-cell recordings from dissociated hippocampal neurons of a rat, and another spike shape we recorded is of juxtacellular recordings. As you can see here, that is already enough to overcome some of MEA's recording limitations, such as signal-to-noise ratio and source resolution. And we could also record inhibitory and excitatory synaptic potentials. The long-term goal of the lab is to implement this method in a behaving animal. This goal is anticipated to be critically impeded by numerous abiotic barriers, such as the intrinsic physical mechanical properties of the implant and biotic barriers. And the main biotic barrier is the brain inflammatory response to the electrodes. That's called the foreign body response. The problem is that those silicon-based rigid platforms that inserted to the biological brain tissue induce immune response. An example of how the immune response looks like is given here. The electrode is pulled out, and you can only see the void in the center and the dense accumulation of cells around it. The immune response is composed of microglia, astrocytes, and probably other cell types. And the loss of recording, decreased quality, and low source resolution in MEA technologies is attributed to this foreign body response. This is usually explained by the displacement of neurons away from the platform, high seal resistance of this thick immune response scar tissue, self-assembled biofouling of the electrode by proteins that increase the impedance of the electrode, and neurotoxic environment in the vicinity of the electrode as a result of cytokines expressed by activated microglia. It's important to note here that despite large number of studies, no correlation between the foreign body response intensity and dimensions to recording quality have been found. And also, without eliminating this mechanical barrier, the configuration in which a neuron engulfs our mushroom or any three-dimensional electrode cannot occur. So now that I introduced the general background and aim, here is the outline of my talk. I will talk about the establishment of a method in our lab a few years ago that enables to analyze the interface between brain parenchyma and our neuroprobes. And I will talk about my PhD, which aims to improve the understanding of the inflammatory foreign body responses generated by the cortical parenchyma to our neuroprobes, and to examine ways to overcome the inflammatory barriers. Based on the results and their analysis, we aimed at providing a roadmap on how to apply the in-cell recordings configuration in freely behaving rats' central nervous system. I will not talk about one of the papers where we revealed gender-dependent differences and vaginal-specific differences in the response to pharmacological elimination of microglia from the brain. As I mentioned earlier, there are abiotic and biotic parameters that control the integration of implant with the brain parenchyma. I will not go into details, but to overcome the abiotic aspects of this issue, our lab have developed a special MEA platform which is composed of polyimide, which has smaller Young modulus than that of a silicon-based platform. Young modulus is simply a measure of material's stiffness. You can see that it is also perforated. Those black rectangles are perforations. It can carry our three-dimensional mushroom-shaped electrodes, which are those little dots here, and the planar electrodes. With the understanding that this method and its interface analysis could be relevant to the neuroscience community, regardless of the successful application of in-cell recordings, most of the manipulation in my study were conducted on a planar electrodes. The advantages of using polyimide rather than silicon, besides having lower Young modulus, is the ability to thin section the platform and the electrodes along with the brain tissue surrounding them, thereby to be able to image the undisturbed interface. Recall that in other studies that could not be done because the investigators had to pull out the implant prior to processing for histological observations, and this act obviously dephased the interface itself, and therefore there is no real information in the literature as to the interface formed between the brain tissue and the electrodes. We began by implanting these electrodes and analyzing its structural integration in the brain and its functional performance. This is an example of the information we can get. We are the first to show this kind of information of the interface formed between brain tissue and the intact platform. So this is the platform. It is labeled by asterisks for clarity. And you can also see here microglia cells labeled by IBA1, which also occupy the pores of the platform. Here are neurons labeled by neon and neurofilament. Astrocytes labeled by GFAP, and a merged image of all of which in addition to nuclei labeled by DAPI. In the first work, we conducted a very thorough investigation of the foreign body response using immunohistological method. I will focus on a very small fraction of the results. For orientation, this is the platform. It is implanted vertically to adult female rat cortex. And those are the horizontal sections which defines the shells around the implant. And here we are looking at the cross-section of a confocal image, and the platform is in the center. We measure the intensity of the foreign body response induced by our platform implant by examining the distribution and kinetics of microglia, astrocytes, and neurons. Microglia are the innate immune system of the brain and the immediate responders to neuroprobe implantation, and the understanding is that their role is to identify the implant, migrate to it, and attack it, and also activate astrocytes. But as I described earlier, other groups had to extract the platform prior to sectioning and disturb this interface. Another problem is that what you see here is the resolution of a confocal microscope image. And only by increasing the fluorescent intensity and co-labeling microglia with DAPI for nuclei, further revealed the vast number of microglia cells attached to the platform. This is an important information most of the research groups investigating platform-brain interface entirely miss. Of course, our observations are supported by quantitative analysis of the immune histology sections. This graph depicts two things: the density of cells as function of distance from the platform, and the density of cells as function of time, which is denoted by the gradient change in color of the columns. The distance from the electrode starts from this yellow background, which is the platform itself, and goes centripetally up to over 200 micrometers away, which is also depicted here and here, with 25 micrometers intervals. Those are microglia in blue, astrocytes in red, and neurons in green. And what we see is that the response to our platform is characterized by a few things. Microglia migrate in the first week after implantation and occupy the pores of the platform. They also migrate and adhere to the platform, and their density reduces spontaneously with time. These dynamics repeat itself also in shells that are away from the platform, but with a relatively smaller intensity. The astrocytes behave in a different way. They too extend branches into the platform pores. But as you can see, the dynamic of increase in astrocytes density is delayed in respect to microglia. Whereas microglia density decreases, astrocytes density increases. Neurons are also affected by implantation of the electrode. As you can see, there is basically no neurons in the pores of the platform. This fluorescent is most likely a background fluorescent of the platform and some neurites that extend into the pores. But the density of neurons is recovered gradually with time. So the foreign body response in our hands is minimal in respects to others, yet in most cases with our platforms, we still suffer from the same well-known fast deterioration of recording quality over time. This is depicted here by the spikes that we can record with our platform as respect of time, and the deterioration of a number of platforms in orange, and viable electrodes in blue as function of time. Correlation between the recording ability of the device and the immunohistological observations led us to assume that this reduced performance of the implant is related to the accumulation of microglia at the surface of the implant. And the next slide shows an electrical circuit model describe our explanation for the decay in recordings. So this is the microglia. And this is the planar electrode. And based on the immunohistological picture that I just showed you, we propose that a still resistance formed by the adhering microglia electrically insulate and mechanically isolate the electrode from its surroundings. This mechanism explains why no correlations have been found between the dimensions and density of the foreign body response and the recording quality. And this is also suggested that approaches to mitigate the formation of the insulating glia could lead to improved recording quality and yield. Moreover, in vivo application of the gold mushroom microelectrodes necessitate removal of the microglia that obstruct the interfacing of the neurons with the electrodes. Immunohistological methods are traditionally used to examine the foreign body response induced by neuroprobes implants. However, in view of the findings, it became clear that electron microscope observations are needed to provide sufficient spatial resolution to support this hypothesis. Our study is the first to examine the interface formed between the platform along with the brain parenchyma with transmission electron microscope resolution. This is an example of the interface formed between brain parenchyma and the mushroom-shaped electrode. So this is 2 weeks after surgery. Here you can see a mushroom electrode that extending from the polyimide platform. And you can see also a microglia cell that tightly engulfed the electrode and isolate it from the tissue. In addition, you can also see here astrocytes cell bodies that located a few micrometers away from the platform. The transmission electron microscope observations confirmed that the assumptions that microglia mechanically isolate and electrically insulate it from the brain tissue. We further discovered that in contrast to claims on neurodegenerative processes in the vicinity of implant location in the literature, we actually saw the opposite. The tissue contacting our platform undergo extensive regeneration. You can see that by the extensions of myelinated axons, here in green, right beside the electrodes, neuron cell bodies that close as 1 micrometer away from the platform, synapses formations, and regrowth of capillaries in the vicinity of the electrodes. Following the confocal imaging complemented by the transmission electron microscope images, this is a scheme that depicts the situation: microglia engulfing the electrode, preventing mechanically and insulating electrically the electrode from the neurons, or neurites and branches of astrocytes that extend into the space between the neuron cell body and the electrode and shunt the signals generated by the neuron from the electrode. In order to set a proper extracellular environment to enable the configuration of a neuron cell body to engulf the electrode, we have to eliminate those microglia that tightly engulf the electrode, to remove astrocytes extensions, and actually also prevent neurites growth that interfere with this interface. And eventually, we also need a factor that will lure the neuron to engulf the electrode. So we had to start somewhere, and a good place to start is the main obstacle. Once the hypothesis claiming that microglia implant interfacings is a major component underlying the electrical insulation of the electrode, we began by searching for ways to specifically target and eliminate the microglia from the game. To do that, we used pharmacological agent called PLX, that I will from now on pronounce as Plex. This is an inhibitor to colony-stimulating factor 1 receptor, which is essential for microglia proliferation, survival, and differentiation. Plex proved to eliminate over 90% of microglia in the brain of mice. Combining the use of Plex in our platform that can be thin sectioned along with the surrounding tissue, we investigated the roles of microglia in triggering, participating, and orchestrating the inflammatory cortical brain response initiated by our platform implantation and as a potential drug to eliminate the microglia cells that adhere to the platform. Because no work has been done on rats, we had to calibrate the use of Plex with our adult female rats. Here you can see a control rat in the left column. Here is a rat that consumed Plex starting 10 days before the surgery. And here is a rat that started to consume Plex 4 days after surgery. And the result is that 95% of microglia were totally eliminated in the rat cortex. And in anticipation of a question, how does it affect the behavior of a rat? I will tell you that this did not affect the behavior of rats nor mice. This removal of microglia from adult female rats' cortex significantly reduced the participation of microglia in the foreign body response scar tissue. Unexpectedly, we discovered a Plex-resistant cloud of microglia in the narrow shell, which is less than 25 micrometers away around the implant surface. In addition, and unexpectedly, we also detected a subpopulation of microglia remained adhering to the platform. Assessments of the functional outcome of our experimental manipulations were conducted by recording the spontaneous electrophysiological activity from freely behaving rats in control and Plex-treated rats. I don't present it here, but the yield, quality, and longevity of field potentials recordings were not improved in respect to controls, in spite of the overall 90% microglia elimination. This is accounted for by the presence of microglia, by the presence of Plex-resistant microglia adhering to the implant surface, and this is consistent with our hypothesis. Another conclusion was that in contrast to what people thought, elimination of microglia did not affect the dynamics of the astrocytes response. So the effect on the quality of recording will be related to the presence of adhering microglia and the branching of astrocytes. To sum up this tryout of using the Plex in adult rats, unfortunately, eliminating microglia cells engulfing the electrodes turned to be a complicated process, and the use of Plex in adult female rats did not improve the probability that a neuron cell body will adhere to the electrodes. But we overcame this problem by using a method in which we improved recording quality and yield. This is yet to be published. Thank you.